Okay, so I'm going to take my mask down. There's a couple reasons why I'm taking my mask down. We are vaccinated. When I say we, I do mean Spencer and myself and the staff here at the Headwater Science Center. We're vaccinated, and we're here on Tuesday. So we're closed today. So off comes this mask because we're open 7 days. When we're open too, we're open on Sundays, but it's from 1 to 5. So that's our current schedule. Now, just before I turn this over to the mad scientist, this is Spencer Potter. And Spencer is a brand new employee here. He's worked here about 2-3 weeks, and we're really happy that he's on staff here and he's going to do our show. So our show is being brought to you because of a generous donation from the Beltrami Electric Cooperative. So, hey, tune in, mad scientist, here he is.
Hey guys, hope you guys are all doing wonderfully, just like me. Today, we are going to be doing, let's get started. We'll be doing a water quality lab where we'll be looking for E. coli in some water samples that we've taken today and tomorrow, or yesterday. So, why are we looking for E. coli and coliform? That's a good question. We're looking for those because they're typically an indicator species of the health of a lake. And we up here in northern Minnesota, that's kind of an important part of our culture, lifestyle. So we want to make sure that we are keeping those healthy and clean. So today, for this lab, for our lab, we'll be using a sanitizing boiling bucket. We have sanitized our pincher or our forceps. We have washed our hands up to our mid forearm. We have a smaller sanitized forceps. We have a sanitized gridded membrane that is small enough to let water through, but not small enough that bacteria like E. coli and coliform will get past it. And we will be using a vacuum assist today to help our samples go through the small porous membrane, and that is basically all of our materials. Oh, we do have our samples. It is in a sanitized sampling bag, which nothing has touched the inside of this bag except for the sample that we'll be testing today. Today we'll be testing a sample from the Bemidji Bridge in between Irving and Lake Bemidji. This sample was taken about noon today, which that is an important thing to touch on, is when the sample was taken. We want to get the most accurate sample possible. So we can't just let that sit around for a week or two, because the microbiology in that bag, in our sample, may change. So we try to get them into the fridge as soon as possible and then run our tests also as soon as possible. So I will begin. I will be taking our sanitized plug, filter holder / vacuum plug, putting it into our hooking it up to our vacuum rig. After that, I will be taking out our sanitized gridded membrane. One important thing to touch on is that I will not be touching or contaminating the gridded membrane, because we want to get the most accurate results as possible. So I'll be taking my smaller sterilized forceps, grabbing these gridded membrane, placing it on the vacuum plug, and then I will be putting on my measuring apparatus. This has magnets in the bottom, so it completes a watertight seal, so that all the water is going to be going through the membrane and not leaking out. This has a this is in milliliters. This is measured in milliliters. So the standard that we use is 100 milliliters per sample. So we will be taking our untouched water sample from Lake Bemidji, in between Lake Bemidji and Irving, entering in 100 milliliters. And it's kind of important to be as accurate as possible, just so we can have a consistency within our samples across the board. We will be saving the rest of the sample just in case there's any mess ups, because we are human. We can't always be perfect. Then, once we have our sample into the measuring apparatus, we will turn on our vacuum to assist gravity. This vacuum is creating a negative pressure in these other containers, which is forcing the water, sucking the water through the now unsterilized membrane. But it will be catching all the necessary bacteria that we're looking for today. So,
it's already sucked through there.
yes, it is sucked through there. We filtered through 100 milliliters of water. So, it's always a very important part of science is to record and label all your data. So we are going to be labeling this as Bemidji River today. So now we'll be taking our sanitize our small forceps again, because they did touch the ground, which we don't want that. We don't want any introduction to any other contaminants. We'll be taking our bacteria filled filter paper off the vacuum seal and then putting it into our agar plate. We'll be entering our bacteria filled filter paper into there, and then because bacteria are living creatures, they need food to metabolize, they need food to reproduce. So that's where the agar comes into place. We will be covering the filtered sample in the necessary nutrients that the bacteria will need to grow to get us a decent result. And then it's easy as that. It will turn out to be looking like this. And now we'll need to let this incubate for 24 hours at preferably 35 degrees Celsius, which is about 95 degrees Fahrenheit, which is also similar to the body temperature of the human. But we can't just leave you like that on a huge cliffhanger. We need to see some results. So we did this lab yesterday so we could have a sample ready for you to show you guys. This is a sample from the river outside of, just below Bemidji, in the water, in the Mississippi River. And as you can see, the red dots are coliform colonies, and the blue or dark colored colonies are E. coli. In this sample, we recorded 217 colonies of coliform and 3 colonies of E. coli. So, and one thing to remember always when you're doing lab is make sure that you're washing your hands before and after, because you are using a live organism. We want to make sure that that we can't let these guys hang around for too long. So we do sterilize the petri dishes or agar plates once we are done with them, just so nothing else happens. We put them in a solution of bleach to make sure that all these cells are lysed and dead. Thank you for tuning in to the water lab with Science Spence, and I'll see you guys in the Science Center Monday through every day, 7 days a week, from Monday through